This program contains graphic images and discussion of medical procedures. Viewer discretion is advised.
So the Orthopedic Trauma Institute, if you don't know much about it, is located at Zuckerberg San Francisco General Hospital. And it's founded around 2009. It's been there since longer, but the basic idea of the OTI is to bring together traumatologists, orthopedic traumatologists, clinicians, as well as basic researchers, and to study trauma, as well as to study the bones, the basic biology of bones, in order to heal them more effectively. And over the last 7 years, the OTI has become a world leader in traumatology, and it's a great credit to San Francisco, as well as ZSFGH, it's very difficult to say, that we have it at this institution here, all led by the visionary leadership of Ted McClough. So today, our topic is going to be engineering the skeleton, which is our main focus today. And when we think about something like engineering, a lot of times we think about things like this. We think about cars, right? We think about things that have design processes associated with them. And in many ways, the body is amenable to understanding it as a machine, right? And this is a long history of thinkers thinking about the body as a machine that changes and that we can model it in a way. And it's amenable because we can think about the skeleton in terms of the way that it adds sort of structural support. It's a framework for the body. It gives you shape. It also serves as the attachment point for muscles. It protects vital structures. It protects your brain. It protects your spinal cord. It protects your thoracic organs. It even protects your internal reproductive organs. So the skeleton is critical. It also facilitates movement, right? It acts as the levers for muscles. It acts through joints as fulcrums. And so we can model it in many ways as a simple machine. And we can use very simple Newtonian physics to understand and model how bones and muscles interact with one another so that we can study things, the reactions between actions and their opposite reactions. So in the first part of today's lectures, we're going to have Dr. Herfat, who is the director of the Biomechanics Laboratory at the OTI. And he's going to be addressing these really basic questions of, what is this guy doing? How is he moving his arm, right? And how are the forces affecting his bones? How does he accomplish these particular tasks? So how do we model this human body as a machine? And something to think about with all the stuff that he's going over is the fact that bones in your skeleton comes in all different shapes and sizes, and that the form is really related to something, and it's related to the function of the bones. It's something to always keep in mind when you're thinking about bones and biomechanics. The other thing we want to think about with biomechanics, I know this is difficult to see sometimes, right, if you look closely, yeah, but this happens, right? And this is trauma. So what happened to this guy? How did the forces affect him? And how can we repair them? These are critical questions that we can use bone biomechanics to understand by understanding things like what are the properties of bone, how do they relate to other things, but then how can we leverage these properties of bone to actually better engineer skeletal repair, to treat the body in many ways as if it were a machine. Now, that's one way of thinking about engineering the skeleton. Another way of thinking about engineering, that's the classical way. Another way of thinking about engineering the skeleton is to sort of remove sort of the hubris that we have when we think about skeletons as machines, right? When we think about labeling, how we label bones and we label skeletons, we label them in a way that makes us think about them as individualized parts. But it's important to remember that skeletons and bodies aren't like this. They're not a Mr. Potato Head, right? You don't have individual parts that you can add and replace easily. What do I mean by that? What I mean is that when you are building a human, when you're building a biological system like a human, it's a highly integrated system. If you think about it, every one of us has gone from one tiny cell to trillions of cells. yet over that whole course has remained functionally integrated and has remained functional and viable, right? That's an amazing accomplishment for all of us. So how does it do that? It's an amazing system. How do you permit function while still growing? This is not something that you see in machines, right? This is not an engineered product like you have with humans. As much as my kids want to believe that there are baby cars, that baby cars then turn into grown-up cars, that doesn't happen, right? But that happens with biological systems. So how is that accomplished? And so that presents a challenge then to us from a bioengineering perspective, because every bone in your body, if you think about it, the bones that you have in your body now took years and years to accomplish their shape, their size, and their form that you have now. And so if you're going to engineer something, you have to take that into account. Now, maybe we could have something like this. We're not quite there yet, fortunately. I think at some level, we can't replace all of the parts with mechanical parts. But we can do something else. As we learn more and more about how the body is created, how it develops, we learn more and more about how we can regrow the skeleton. And that will be part of Dr. Chelsea Bahney's second part of this talk, thinking about engineering not as this sort of unnatural engineering, using a human intellect to build a body or build parts, but using the information we know about how bodies are put together to actually leverage that information to utilize the same kinds of information we have there to build new parts. And we can do this because as we learn more about development, the cues, both chemical, biochemical, as well as physical, that drive development, the more we know about those, the more we realize that these kinds of same cues are ones that lead to things like disease, and they're also the ones that are recapitulated during the repair process. So the body in many ways already knows how to repair itself. Can we leverage that to produce sort of innate healing? In other words, can we use it to build new bones? Can we use it to build new tissues? All right, so with that, I'm going to turn it over then to our first speaker of the night, Dr. Safa Herfat, who will be talking about bone biomechanics and going into a little bit more detail about some of these issues. Thank you.
Okay, so today we'll cover the mechanical function of bone, its adaptation to loads, types of mechanical loading, the mechanical properties of bone, the causes of fracture, fracture risk factors, and then some of the research that we do at the biomechanics lab at the Orthopedic Trauma Institute. So basic mechanical function. I don't want to get into too much engineering here and biomechanics, but the skeleton plays a critical structural role in bearing functional loads. So these are weight-bearing and muscle loads. You can see by these first two diagrams. The second one is a free body diagram. We use this a lot in mechanics, particularly in biomechanics, to basically map out all of the loads, and it helps aid us in our calculations to figure out the balance of the loads. So you can see here when we're calculating the hip force, so the resultant joint force. So how much force is the hip seeing? This is important to us in the lab because when we're testing a femur or a fracture fixation of the femur, a fracture fixation strategy with my orthopedic surgeon colleagues, we need to know how much force should we apply to apply a physiologic load onto the joint. So we know that the abductor tension, the muscle tension, creates force across the joint, and so does the body weight. In this balancing act, just during single leg stance, you'll see that the joint will see about 3 times body weight. And during gait or running, you can see up to 4 or 5 times body weight. So the other thing that the bones do is act as a lever or a rigid body to help our musculoskeletal system perform a certain task. So if you look at the diagram to the right, you'll see that if you want to lift the ball, you can only do that with the muscle tension if the forearm piece is a rigid lever. If it begins to deform, you're unable to lift that ball. So the structure actually adapts to the loads. There are quite a bit of structure-function relationships in biomechanics. Here you can see that the trabeculae, or the spongy bone in the proximal femur, so again, we're talking the hip, aligns itself to the loads that the hip would see. So if the joint reaction load is coming down here, you'll see that the trabeculae actually align itself to that compressive group. Well, what else happens biomechanically? If you're applying this compressive load, you also have a moment arm here, and you're creating a bending moment across the femoral neck, which creates a tension up here at the top. So you'll also have the trabeculae align in that tensile group. And so when the bone sees strain, the trabeculae can grow. You can increase the number of struts. You can increase the thickness of these trabeculae, the connectivity, and the orientation. And in addition to the spongy bone, you also have the thick cortical bone in the midshaft of the femur. And this also can thicken due to loads. So here you see a tennis pro, and you can see that the humerus on his right hand, which is the arm that he uses to swing his tennis racket, is 50% thicker than the other. So the bone will adapt to the loads that it sees. So what kind of loads would a bone see? So let's talk basic mechanics. What are the different types of simple loads? And again, these are simple. Your bones may see complex loading of multiple of these loading structures or loading regimens at once. So the first and the most basic would be a compression force. So you're compressing down, and here we use a cylinder to model a long bone. So in compression, you're basically smashing down that bone. With tension, you're stretching it. You can imagine a rubber band. And then bending is actually really interesting and is something that the orthopedic surgeons really have to think about when repairing fractures. So here in the bottom, you'll see that cylinder, or think of the femoral shaft, your mid femur, is bearing a bending load. So on the top, you'll actually see tension, and on the bottom, you'll see compression. Now later on, we'll learn why this is so important. Also, you can experience shear. So you can think of two sliding surfaces and shear, and then torsion or twisting. So basic bone mechanical properties. We hear words like strength and stiffness all the time. So strength is basically the load that the bone can bear before breaking. So when we hear things like your bones are weak, then their strength, the ultimate strength, is low. So here in the diagram, we see a load versus displacement chart. This is a very simple chart of a bone taken to failure, and it can be either one of these three loading modes. This is just a representative chart. So you can see the load will increase as the displacement increases, and initially it's a linear region. This linear region, the slope of this linear region, is what we use to calculate the stiffness. So later on, as it increases 1-to-1 ratio, and you have this linear stiffness, it reaches a yield point. And after this yield point, you enter the plastic deformation region. We'll talk about this some more. So the strength. What contributes to this strength? So you have the total bone mass, you have the structure, the geometry or the architecture of the bone. I showed you some of the trabeculae and the trabecular structure in the proximal femur. And then you have the material properties or the tissue composition. So stress and strain, these are all their basic mechanical terms that we use. And these, think again, force and displacement, but now we account for the geometry. So here we have a force applied to an area, and stress is just force over area. So it's force applied to the area, and then strain is the deformation or the change in length or change in shape. So I showed you a force displacement curve, now I'll show you a load deformation. Pretty much the same thing. So you have the linear region, you reach a yield point, and then it starts to give until you have a fracture. And this area under the curve is what we call the energy, or energy absorbed, or fracture toughness. So we have the yield stress, the ultimate stress, which is the max stress or load that the bone can bear. And then this was a study that compared normal to osteoporotic. We'll talk about osteoporosis some more. So you can see that the osteoporotic bone samples, in this study they tested it in tension. You can see that it failed at a lower stress, so lower ultimate stress. And then the stiffness, or modulus of elasticity, was less. And then the area under the curve, if you assume that it failed right there, and then the normal failed right there, the area under the curve, or the fracture toughness, was less in the osteoporotic group, which contributes to the brittle nature of the osteoporotic bone. So bone is also anisotropic. What do we mean by anisotropic? So an isotropic material has the same mechanical properties in all directions. Metals are a good example of this. Bone is anisotropic, so it's dependent upon the direction of loading. Here on the chart below, you can see that these are representative strain gauges. So you're measuring the strain in different directions. And you can see the stress strain curve. You can see based on the direction that it's aligned that the stress strain curve will differ. So the mechanical properties will differ. So this is attributed to the specifically oriented components in the microstructure of the bone, which you can see here. And bone, again, I mentioned tension and compression and shear as well. So bone is actually very strong in compression. If you think about the lower limbs, we talked about the femur. When I'm standing here, for instance, my bone is under compression, and that's a very common loading that it experiences. Bone is not as strong in tension. This is something that the orthopedic surgeons have to take into consideration when they're healing a fracture. And then bone is very weak in shear. So viscoelasticity, what does this mean? The stress strain character is dependent upon the rate of applied strain. So again, a stress strain chart, you'll see that if it's loaded fast, you're actually picking up higher ultimate stress, higher stiffness or modulus of elasticity, and greater area under the curve or fracture toughness. So it's time dependent. So in bone and other musculoskeletal tissues, it's viscoelastic because it has two different characteristics. It has a viscous and an elastic. So the viscous is predominantly due to the fluid flow that you'll see. So there's actually fluid moving around in your bones, and it actually contributes to this mechanical response. So at a higher strain rate, you have higher stiffness, higher strength, and higher energy. Okay, so what about when things go wrong? We have bone fractures. So here are some simple fracture patterns. And again, you see the loading modes across the top. So we have tension, compression, bending, and torsion. So these loading modes usually lead to these fracture types. So in a tension loading mode, you'll see a transverse fracture. Compression, you'll see oblique. Bending, you'll see butterfly. And torsion, you'll see spiral. So what are the common causes of fracture? There's three. Trauma, or traumatic fracture, fatigue, or overuse, and pathologic. So a traumatic fracture, you can think of acute high energy. So in this case of I believe it was a 17-year-old patient falling out of a tree, you can see a compression fracture of their vertebral body in their spine. In this case, the load exceeds the strength of the bone. And here, again, going back to the hip, you see another high energy fracture. So let's talk about some common examples. So we had a great year in Tahoe this year. I'm sure some of you have gone skiing or snowboarding. But let's talk about a ski boot fracture. Say we're going down, flying down the hill, and our skis get caught in something or we hit something, and we fly forward, our bindings malfunction, they don't release, and we have a fracture. So what do you think is causing, what type of loading do you think you would see on this bone? Okay, so bending. That would probably be my guess. And why? So if I draw it out a little bit, we'll say my ski gets stuck as there's a lot of energy involved. I'm flying down this hill, and I have my ski boot, and my tibia, my shin, contacts that, but all of my momentum is still going forward. So all of my body weight, all of my energy is still flying forward, and that can create quite a bit of bending on that tibia. And so tibial fractures are very, very common in skiing and snowboarding as well. And 90% of them, estimated 90%, are actually non-contact. And what that means, you're not running into anybody. Okay, so another one, distal radius. So high energy again, let's say I'm flying down a hill on my bike, and I fall off with an outstretched hand. What do you think's happening here? Distal radius, right here, next to the wrist. I heard it. Compression. Yep, that's a great guess. So again, high energy, the loads exceed what the bone can handle, and you'll have fracture, as you can see here on the x-ray of the distal radius. Okay, last one, clavicle fracture. So this time, again, high energy. I'm riding my motorcycle on the highway, and I slip and fall off and land on my shoulder, as you can see here. What do you think's going on here? What kind of loading? Yeah, compression. It can be shear as well. So this one's kind of tricky because the clavicle is S-shaped. So that can affect, and the way that you fall can affect the loads that are applied. So in this case, if we assume that the load is directly upwards, you can assume it's compression. But again, we're talking very simple fracture patterns. It can be a mix of some of these bending modes or loading modes. So here in the x-ray, you can see an oblique fracture pattern. As I had shown before, that is usually due to a compression. Okay, so stress fractures. These are overuse fractures or hairline, fissure, fatigue-induced fracture. So fatigue, I'm sure most of you have heard the word muscle fatigue. So it's a result of accumulated trauma from repeated loading. And this is something you'll see commonly in runners or athletes that are jumping over and over and over again. These are not accidental loads. Here you can see an x-ray of a stress fracture on the foot and on the tibia, the lower leg. Okay, so pathologic fractures. These are caused by a condition or disease that led to weak bone. Osteoporosis is the most common. In these cases, you don't need high energy. These are actually low energy, which is defined as falling from a height of standing or less. In this case, a very frequent mechanism is just falling to the side. And you can see here by this diagram that the mechanics, the loading applied to the femur from what it's usually used to seeing, and this compressive force coming down on the femoral head, changes now because you have a force as it hits the ground. You have a reaction force on the greater trochanter. You're applying your body weight down on a different area of the femur, and so the area that was used to seeing compression, that was built for compression in a normal activity of daily living, now can see a high tensile stress at the bottom of the femoral neck, and it's not used to bearing that load. So you can see a fragility fracture there, as you see on the x-ray. The other conditions that can cause this are cancer, infection, bone disorders, or a bone cyst. So here is an example of bone tumors of the femoral shaft and humeral metaphysis, or proximal humerus. Okay, so what happens when we age? Your bone mass decreases, your strength decreases, your bones become more brittle, and this can increase your fracture risk. Your bones can actually remodel due to, or basically compensate mechanically. And you're shifting the effective bone to the periphery. So what does that mean? You have your young bone here, and as the inner bone starts to resorb, on the outer side of the bone, the outer periphery, new bone is formed. This actually increases the moment of inertia as the cortex, the total cortex, thins. And this increases the resistance to bending and torsion. So your body, again, can adapt to the loads. So osteoporosis, I'm sure you've all heard of this. It's a disease characterized by low bone mass and structural deterioration, increasing bone fragility and fracture risk. Greater than 25% of women 80 and older have been reported to have osteoporosis, and leads to about 9 million fragility fractures. Most common in spine, hip, and wrist. So what are some of the factors that can increase your risk of fragility fractures? I have them all here, but I want to focus on this one. Physical inactivity. Weight-bearing exercise is good for your bones. So in the lab, we actually perform bone density tests when we're screening the test specimens that we use for our studies. So if I'm doing a fracture fixation study, studying repair strategies for fracture that we would usually see in osteoporotic patients, we'll actually screen with a DEXA scan. And this is actually the clinical standard you can see here. It's basically an X-ray, and it measures the bone mineral density at either your hip, your spine, or your forearm. So these are examples of the outputs, and based on these, we have a T-score. And the WHO gives ranges for normal, osteopenic, and osteoporosis based on the T-scores. There are other assessment tools for fracture risk as well. This one was developed in 2008 by the WHO and takes more factors into consideration than just bone mineral density. So the research that we do at the Biomechanics, and we can't wait till you all come visit, is mainly experimental testing. So we'll actually take bones, we'll work with the orthopedic surgeons, and implants, they basically perform the same surgery that they would perform on a patient. They'll do it here on a bone. And we'll compare different fixation strategies mechanically. So we're doing a biomechanical evaluation. Sometimes we'll use artificial bones as well. And we're applying, again, physiologic loads. We need to know what these bones will see in normal activities of daily living. And those are the loads that we apply in the lab setting using these mechanical testing systems. And we can measure the strength, stiffness, and fatigue life of these constructs and of native bone itself. So the other thing that we do is finite element analysis. And this is more on the computational biomechanics side. So we can do the same thing that we do in the lab, but virtually. We can virtually create a surgery and then simulate the mechanics. And this allows us to get a heat map. Basically, there are many things you can output, but the most common is a stress map of what's going on across the entire construct. I'd like to thank you, and Chelsea?
Thank you all again for coming. And actually, I was part of the first course, and I do see a few familiar faces from the first time the OTI did a lecture series. So hopefully this is a different topic, and hopefully it will provide a different perspective on what both Nathan and Safa introduced, which was this idea of the engineered skeleton. And they talked about how form and function are really critical to what our skeleton does, and how the body naturally develops and has evolved to have a skeleton with the specific form and function. And our bodies do have the ability to repair or regenerate themselves, but not always. If the injury is too big, or if we have some sort of disease that we get, sometimes you won't be able to repair or regenerate your skeleton. Unfortunately, our skeleton does not heal as well as we age. And so the goal of regenerative medicine is really this idea of, can we capitalize on the innate regenerative capacity of bones and cartilage in our skeleton, to learn from it, but also to accelerate it or use it when we don't have, when that natural tendency isn't there. So who knows what this animal is? Ted? Ted? There you go, the winning prize in the back is the axolotl. So we do look to the axolotl as inspiration for regenerative medicine. So this is one of the few species, Nathan, correct me if I'm wrong, who can regrow an entire limb. And not just heal it with scar tissue, but actually regenerate a full limb. So this is a picture. If you chop off the leg of this axolotl, it will slowly regrow. The fingers will reform, the muscles will come back, the vasculature will come back, it will fully regenerate. There are some things, lizards and salamanders' tails have the ability to regenerate to some extent, but it's more of a healing response, not this full regenerative response. So the axolotl we can use as an example to start to study the molecular, cellular, and genetic mechanisms that are regulating regeneration, and then we can try to capitalize on those principles, those basic principles for human regeneration. So this is a little bit what Safa presented, and this is how we fix things now in orthopedic surgery. So fortunately, we are the basic research side, and so hopefully without offending anybody, I can say that orthopedic surgeons are very, very talented carpenters. They have a toolbox. You should see the OR when they are operating. It has drills and hammers and saws and pins and nails, and it's a pretty elaborate process of trying to put these bones back together or trying to fix a joint. But a lot of it is really still biomechanics. So probably some of you have either had this or know somebody who's had a total knee replacement. So you can see here what's happened is that we've lost the cartilage. Cartilage doesn't show up on x-ray, but cartilage keeps this space and it keeps your joint lubricious and allows you to articulate your knee. When you start to lose your cartilage, eventually you get bone-on-bone articulation. So cartilage is interesting. There's no blood vessels, there's no nerves. You don't even feel this process until it's come to here, because this is when you hit the bone and now the nerves hit and you start to feel the pain of the joint. And at this point, there's nothing you can do. And so you end up getting a total knee replacement. This is a titanium alloy joint replacement. Maybe some of you have had a foot or toe or ankle fused, but that's another thing we do when the joint isn't working anymore. We just start to fuse the bones together. So you can see there's these really elaborate plates that have been developed, and there's been huge advances in orthopedic surgery because we figured out through biomechanics how to make these plates stronger, how to make them adapt to different bones. In a fracture, this is a fracture. You can see there's all these little pieces. This is a really big challenge to put back together, and so they use these bone plates. I'll talk about later, but this bone has a very low chance of healing. So despite all this, putting it back in together correctly. So the idea of regenerative medicine is how can we restore form and function of diseased tissue through biological processes instead of just these mechanical and hardware processes. So the FDA has defined regenerative medicine to include a pretty wide range of topics, and I'm going to not hit on all of them, but try to introduce very briefly each of them so you just have an idea of what this might mean in orthopedic surgery. So regenerative medicine could just be transplanting human tissue. So this could be living tissue, we call that autograft, take bone from one part of your body, transplant it to another, maybe bone from a part of your body that's not load-bearing, and so you could spare it, so to speak. You can also get dead tissues, so allograft tissue. And so this is still classified as regenerative medicine because you're using the biologics. And it's probably the most simple form. The next one is cellular therapy. I'm going to cover this in less detail. This is a topic we talked about in our last mini medical school as specifically stem cell therapy. There's also this idea of tissue engineering, and we're going to talk quite a bit about tissue engineering and this idea of maybe regrowing an entire organ and how you might do that. Gene therapy. So if you have a disease that is caused by a particular mutation, can we go in and edit that particular part of the gene so that you can take out the bad DNA and put in better DNA? And so this is not as active in orthopedic surgery, so I'm not going to talk about it too much. We don't have that many diseases that are causing common diseases that are causing problems, and so regenerative medicine hasn't really gone in orthopedics to gene therapy, so I won't cover that too much, but there are a few diseases that we've been considering, but most diseases are caused by these mechanical traumas that Safa introduced. And so the last one is just, can you inject something that will stimulate endogenous repair? So can you trigger the body to heal itself? Okay, so this is the joint I told you, so really our quest is trying to find a biologic solution to this. So the challenge, particularly with cartilage, is that cartilage has no innate regenerative capacity. You injure it, you're done. This is really what inspired me into regenerative medicine, because I was a ski racer and I've done this. I've torn various, who knows, I don't want to know, parts of my knee cartilage, and I'm going down this path because really once you start to degrade it, your hole just gets bigger and bigger. Because as Safa was talking about, when your joints articulate, it really requires that all of this cartilage is distributing the load evenly across this joint. And you can imagine that if you get this hole, all of your biomechanics are going to be thrown off. What that starts is this accelerated remodeling process of the cartilage that the body has no ability to recover from. So slowly your cartilage just begins to degrade. So we want to know is, could we try to stop this process with something earlier so that you don't end up with a joint replacement? So I want to talk about some of the current techniques and then talk about some of the future techniques. So one of the current techniques that's more or less new, but it's this idea of transplanting living tissue, is taking cartilage from non-load-bearing areas of the joint and transplanting it into that hole, and it's called a mosaicplasty. And so you can see here, it's definitely an art for the surgeon to develop this. And what this mosaicplasty does is it improves the biomechanics. And so now you can hopefully restore the biomechanics so that you can stop the biological degradation that's happening. But it doesn't really trigger any active repair of this cartilage. So we've made some progress, but still not quite there. Has anybody had a friend or themselves had anything injected into the knee to try to help knee pain? See a few hands up. So one of the things you may have had injected was hyaluronin or hyaluronic acid. I'm sure there's fancy names for it. It's essentially egg white. But it mimics what is the synovial fluid that provides all the lubrication for your joint. And so as this cartilage starts to dry up, you don't have this lubricious surface. And now instead of having oiled metal rubbing on each other, it's not oiled, and so this is what that does. So people can go in and have this injected. The nice thing is it does mimic this synovial fluid, and it provides temporary relief, but it probably lasts 3 weeks, a month, maybe 2 months. I'm not exactly sure. Probably is different for anyone. And it doesn't activate any repair. But it is a mimetic of a biological process that helps to provide relief. Another one is called platelet-rich plasma. And so this is a picture of blood. You can get your whole blood, you spin it down, and you can separate out this plasma from the red blood cells. And so people have started injecting platelet-rich plasma. It's become famous by various athletes having this injected. And there's various stories of this helping, and it could help. It certainly may stimulate repair in some situations, but the results are highly variable. And the biology of this is not very well characterized. And right now there's some really great research going on to understand what is in this platelet-rich plasma and why it helps some patients and not helps others. I think what it depends on is what your biology is and what your injury is. So for example, if you have a lot of a protein that stimulates blood vessel invasion or angiogenesis, that wouldn't be good for cartilage repair, but it would be great for bone repair. So people are working to try to identify what is in this platelet-rich plasma, how it differs between all of us, and then in what cases it helps and what cases it doesn't. And we're going to need really large data sets before we can start to understand and characterize to make generalities about what this might be. And then eventually maybe we could come up with a fully synthetic version that's good for each application. So the other one is getting stem cell injections. There's lots of different sources of stem cells, and in the last lecture I really tried to define what a stem cell was. But for the purpose of this, it's just a current technique in regenerative medicine. People will have injections of stem cells. And this is a picture of a mouse that got a systemic injection of stem cells. And all these, this is a heat map, and basically it shows you how many stem cells were injected. There were tons, they were all over. And within at day 0, they were everywhere. They get localized to the lungs because the body, everything ends up going through the lungs, all the vasculature, so it gets filtered through the lungs. But at 3 weeks, there's nothing there. And actually, the data supports, it's different for different tissues, but about 99% of the cells you inject are gone within 24 hours. So there really is minimal cell engraftment. These cells aren't going somewhere and just engrafting into your cartilage and growing new cartilage when they're injected. But they do have a benefit. And we're still trying to understand what exactly that benefit is. But it turns out this stands for mesenchymal stem cells. So these stem cells actually provide a number of what we call trophic effects. So that basically means they don't actually engraft, but they are supportive. They are anti-inflammatory. They are, so they suppress inflammation. They, can't read this here very well. They're anti-fibrotic, so they prevent scarring. This is inhibit apoptosis. They prevent cell death. And then they also trigger innate repair. So while they might not go in and grow new cartilage for you, they do have a supportive function. And so people are working to characterize this process better. So what is the future of regenerative medicine? I think that we've started to get in the clinic, we have started to address some of the major problems, but our real goal is to restore the full form and function, and we're not there. And the problem is that skeletal tissues have both a biomechanical and a biological function. So it becomes more complicated than trying to just stimulate repair or mimic the biomechanics. So tissue engineering is a strategy that we can use where we take the cells and then we create a scaffold for those cells, and then we put that entire system in that really is a replicate of the organ or the tissue that you're trying to replace. So one of the technologies is this one that I'm going to introduce called a hydrogel. And this one is advantageous because you basically start with a liquid, and we call this liquid a monomer because you mix the cells and this liquid together, and then you can shine light on it, and it polymerizes into this hydrogel scaffold. So this is a lot like the technology they use for contact lenses. It's also not that far from making Jell-O. And so, but it's a very biocompatible system where we can activate the gelation in about 30 seconds by shining a bright white light directly on it. And we've developed a whole bunch of systems so that this process is entirely cytocompatible. So we've been making plastics and those sorts of things using similar technology for decades, but it turns out all of the things you use to gel these process, or originally used to gel these scaffolds for plastics, is not compatible for cells. So we had to re-engineer everything so that it can be done at biology temperature, so your body's temperature, so that it can so that it won't kill the cells, so that the chemicals in there aren't toxic. The other advantage of this system is because it is a liquid before you shine light on it, you can imagine that you could just deliver it laparoscopically right into the defect and shine the light through one of those laparoscopic holes, and now you can fill that particular defect, and hopefully you could go in, you know, right after you tear your cartilage in a ski injury or ski accident and not have to wait till your very late stage. Another cool technology is really, instead of filling a focal defect, this idea of creating the whole articular cartilage surface. So what's interesting about this scaffold is they use this technology I'm going to talk about, electrospinning, where they basically wove together very strong materials, and then they shaped it using, it's a 3D biodegradable scaffold that was woven, and then they molded it to be the shape of a joint. In this case, it was a hip joint. And then they seeded it in the lab with stem cells. They grew it in a bioreactor, so in the lab for a couple weeks. And then this can now be popped over the bone, and you have a new articular cartilage surface. So that's the future. We're not there yet. Now I'm going to move into bone. So kind of a different set of problems, but also still skeletal tissue, also needs regeneration. The advantage of bone is that bone actually has a good innate response. How many of you have broken a bone? It's about right. So there's about 15 million fractures a year. 60% of us have broken a bone by the time we're 60. For those of you who haven't broken a bone by the time you're 60, I broke at least 10, so I cover quite a few of you. Again, why I got inspired into regenerative medicine. But there's about 50 million fractures a year, 1.6 million trauma patients, 1.6 million bone grafting procedures, and 10 to 20% of fractures don't heal under normal conditions. Which doesn't sound like a very high percentage, but when you have 15 million fractures, that becomes a pretty significant number. More importantly, 47% of fractures with what we call comorbidities won't heal well. So a comorbidity is a complication. Unfortunately, the complications are pretty common things like getting older, smoking, diabetes, being overweight, damaging the vasculature, or maybe to somebody's question earlier, the muscle. If you have a really big muscle loss associated with the bone loss, the muscle can contribute biologically to the fracture healing. So not just mechanically does the muscle play a role, but biologically things from the muscle. So if you have any of these comorbidities, now you have this chance of fracture not healing going up to 47%. And this is an example of one of those fractures that the surgeon later had to go in because it didn't heal at all. And you don't know it doesn't heal until 3 months later. And then they say, oh, let's wait and see if maybe it just needed a little bit longer. So then 3 months after that, you go in. So this is 6 months before they determine that this is a fracture that's not going to heal, and they're going to go try to fix it again and add bone graft in, or fix it with new plating techniques. So the challenge is that bone isn't as simple as cartilage. I don't know if you remember that I told you cartilage is a single cell type. No blood vessels, no nerves. So we started trying to engineer cartilage because it seemed like the easiest engineering problem. It was a very simple system. Bone is a more complex tissue. It has bone cells, it has blood vessels, it has nerves, it has marrow space, and all of these have a functional role. So how do we regenerate all of those things, kind of like our axolotl? So the current techniques in regenerative medicine for bone are actually pretty basic and have been around for decades. It's really just using bone to fill this defect. So this is an example of an external fixation. And here's a defect that's too big to heal on its own, so they shove in a bunch of bone graft material. You can take that bone graft from the iliac crest and transplant it. The problem is about 20 to 40% of people have pretty significant pain from where you took the bone. Or you can use this dead bone, allograft, and transplant that in. The nice thing is there's tons of supply of that. The bad part about that is it really doesn't work that well. It doesn't vascularize, it doesn't integrate, and so there's some pretty significant problems with that, that technique. So at the OTI, we have thought about this problem quite a bit, and we've really felt that one of the reasons that bone grafting might not work is that we aren't taking this approach where we're trying to model what the system naturally does during development or repair. And so current technologies to fix bone use bone. But bones don't develop as little tiny bones. They actually start as cartilage intermediates. And also, bones heal through a cartilage intermediate. So this is a picture of a mouse bone from our lab that we broke, and this red stain is cartilage. So you can see in between the two bone ends, you get this big cartilage callus. And the purpose of that cartilage callus is actually to rapidly stabilize these two joints, but maybe not to fix it so well that you don't keep walking on it and you actually sit down and let yourself heal a little bit. And then this cartilage becomes calcified, gets harder and harder, and gets remodeled into a fully vascularized bone. So when a fracture fixes, it creates a bone that's completely indistinguishable from the injured or the original bone. And so we knew that if we could just capitalize and completely recapitulate this process, we might have a very successful healing opportunity. So this is a technology we call developmental engineering. It sounds fancy, but it's really just logical. It's engineer a system that models tissue development or repair. And so we've been working on this idea of indirect bone formation. So take a stem cell, and right now you can make cartilage and bone from these stem cells. So traditionally, everybody that makes cartilage is trying to fix the articular cartilage that I talked about earlier. If you make bone cell, you're trying to fix bone. Seems logical. But one thing that we could do is actually promote this process of bone formation, which cartilage becomes this big, what we call a hypertrophic chondrocyte. This hypertrophic chondrocyte is highly bioactive. It makes all of the proteins that we need to trigger the blood vessels to come in and to trigger the cartilage to turn into bone. So can we transplant these chondrocytes into the bone defect and actually create bone indirectly, but would that bone be a better quality bone? And so we did a study where we implanted cartilage, we implanted the living bone, so the autograft bone, and we implanted dead bone, or the allograft bone, and we compared the healing outcomes. And what we found is that cartilage performed as well as what I'm calling here isograft, but it's that idea of the living bone transplant. And it integrates as well mechanically, so we used a very miniature version of those strain stress devices that Safa has for testing human bones. We have a mini one for mouse bones. We tested that. We did just as well with cartilage as with the bone allograft. And the interesting thing is the bone mineral density, if we do something like a DEXA scan, or in this case it was a micro CT to measure bone mineral density, even though we started with cartilage, we'd eventually get the same bone mineral density as the bone. So going through indirect healing can provide as good a results as living bone, which does pretty well. We don't have a lot of living bone to spare. So this is the picture of the injury when we started. This, you may notice, looks exactly like the human version. So we have everything miniaturized for our mouse studies. And then 4 weeks later, after we put the cartilage in, we can see new bone forming. It's highly vascularized, so all of these spaces between the blue is where bone marrow space is, and all this little black stain, I don't know if you can see it, but that's the blood vessels forming. It also is highly integrated with the host bone. So we believe this technology, or this approach of taking a developmental engineering approach, has solved two of the main problems in bone regeneration. To scale this up, we need to figure out a way to do tissue engineering. And so the building blocks of tissue engineering are the cells, the instructive signals that tell these cells what to do, and then the three-dimensional scaffold. And I'm going to talk just briefly about each of those. So the components of tissue engineering. So I don't know if any of you saw Mission Impossible, but our difficult mission is trying to figure out how we can regrow the skeleton. And here are our components, going back to the housing, which is the scaffold. How do we trigger healing? And what cells can we use? And which cells? What kind of stem cells do we use? Unfortunately, we really have not settled on anything. There's a ton of research. This is a really active area of research, but there's still pretty fundamental questions about what scaffold material do we use? What factors do you need, and how do you deliver them? What cells? Where do you get those stem cells from? And so just want to give you kind of insight into where the research is going and what the next generation of tissue engineering is. So this is a cartoon of the cell. And at a nanoscale, what those cells are seeing. So the first thing it's seeing is this housing, this scaffold. And so this scaffold needs to be biodegradable. We don't want to put in some synthetic system that never gets remodeled because, again, our goal is to create a fully functional tissue that's really indistinguishable from the original. This scaffold also can have physical properties so that its strength, the porosity, the hydrophilicity, so how much water it holds in, are all really important qualities of your tissue that we have to engineer into it. These little green dots are actually what's talk to the cell. So the cell will adhere to these matrix molecules, and that's how force is transmitted. So the cells can actually sense force by touching the scaffold and interacting with it. And lastly, these pink dots and even these right here are the idea of bioactive factors. So can we put growth factors in? So when you put the stem cell in, the stem cell knows what to do. So we now have this toolbox of techniques in tissue engineering. And really, this is our toolbox now instead of, is the polymer synthesis. And so most of these are kind of synthetically organized. But what's nice is we have techniques now that we can individually engineer each of these components and try to understand what is most important for regeneration. So briefly, there's ways to think about what this scaffold should do, and we can think about natural and synthetic components to it, and what are some of the pros and cons of those. So the pro of the natural system is that it's biologically active, obviously, and it's biodegradable. Synthetic systems, we have a lot of control over. We can control every one of these aspects because we've understood the molecular structure of them. The cons of this system is that the natural often are pretty weak, so they don't, by the time you manipulate them, they become weak. If we transplant tissue from one person to another person, there's always a concern about immunogenetic response, and they're hard to modify. So the con of the synthetic is that they're typically inert. The cells don't know how to interact with these essentially plastics, and the cells don't adhere, and the cells don't do things like proliferate on them. So really, our idea here is we want to combine both the natural and synthetic. So tissue engineering is going towards what we call these biosynthetic hybrid scaffolds. And so in the last few minutes, I just want to show some pictures of how people are working on this. So photo patterning, this is some work done at UCSF, where people are studying how the pattern can influence, this nanoscale pattern can influence regeneration. So you can see that these little pillars can be different heights, they can be different spaces apart. You can have holes here. We know that these holes help trigger vascular invasion. These pillars help with cell adhesion and cell proliferation because they mimic some of the strains and stresses that those cells are seeing naturally. So we can recapitulate this with synthetic environments, and we can stack these things up, and so that we can build big structures that could eventually go on top of your cartilage or go into your bone, and cells can be seeded throughout this whole system. Another one is electrospinning, and so you can do that's 3D weaving or electrospinning by basically taking a polymer solution and at a pretty high speed sending it out of just a standard syringe. You put a high voltage across it, and this ends up creating a fiber mat, and basically it looks like little fibers, like your collagen fibers that make up bone and cartilage and tendon. So all of the building block of that scaffold you can make through this electrospinning, and now people have gotten fancy, so it's not just this random fiber mat, but you can actually orient this and weave it together, much like a quilt. And the other one is bioprinting. So we have a 3D bioprinter in the department that we're working on, and we can do things like put different cell types in there and build organs with multiple different types of cells. We can have one cell solution and one polymer solution, and we can layer them and essentially build little shapes that we want to test and see how the interactions between those material or cells work together. So that's a little bit of an insight into the future and what the research lab is working on while the clinic is trying to capitalize on the tools that they have available. So our goal is to regenerate. We're not there. People always ask me, excuse me, how far we are, and I always say 5 years. The caveat is I've been saying that now for about 15 years. So we still have a ways to go, but I think we're making exceptional progress, and I'm happy to take some additional questions about what you're interested in. Yeah, the question was if any of these techniques are in prime time, and no, none of these techniques are in the clinic, but people have taken aspects of them, and aspects of them are in the clinic. And so, but I do still hope that we're closer to 5 years away than 20 years away. So hopefully within our generation, for sure, we'll see some of these technologies, because technology is changing so much more quickly now. So it's a little bit about what Safa was talking about, even cartilage. So the question was, why could runner's knees, even after all that loading, maybe still be better or more healthy? And so the reason is that cartilage actually, the anabolic response, or the pro-regenerative response, is triggered by mechanical loading. All of the skeleton needs mechanical load to function. So the constant motion that you're putting on these things helps the cartilage cells stay healthy and make that hyaluronic, or the synovial fluid and the hyaluronic acid that it needs to be lubricious. So the caveat on maybe why it's not all runners' knees is I think the runners who have managed to do it for a really long time have really good biomechanics. Their joints are really well aligned. They have good form when they're running. So if you have poor biomechanics while you're running, you could actually end up having it be detrimental. So there is a fine balance, and I think some people are built for running, but the loading is definitely good. So the worst thing you can do for your knees when it's hurting is stop moving. If you can't run, biking, swimming, things that are maybe less impact but keep your joint moving so that you're making the right molecules to try to keep your joint healthy are key. Yeah, so I didn't bring that up, but we actually have little mechanical stimulators in the lab where we can load those constructs to try to understand what the forces are. Unfortunately, it becomes really complicated engineering. As Safa was describing, you've got shear, you've got compression, you've got tension. It's pretty hard to make that machine and control all of those components. The body does a much better job. So we can study how compressive forces contribute to healing, but it might not be the best model of the system or of the body because you aren't considering all of the strains and stresses at the same time. Yeah, the question was, where are we on stem cell injections? So stem cell injections are being done now. The caveat is maybe that, I guess there's a few caveats, and maybe just a word of warning. I think we don't know enough about them yet, that you just need to make sure it's not controlled by the FDA. So sometimes the process of getting the stem cells isn't maybe going through as rigorous practices to isolate them well. I'm not sure there are, not all surgeons are trained in how to isolate stem cells like we do in the lab. And so are you really getting stem cells? That's the first question. The second question is, people really think that the the way it's sold is that these stem cells are going to be injected and they're going to go make a new bone or make new cartilage. And so to the point earlier is that they might be helpful, but they probably aren't helpful for everyone. And if they are helpful, it's not because they regrew your bone. It's because they triggered that endogenous healing response and knocked down inflammation, which is a big reason people feel better after stem cell injections. I don't know if I have a good answer. Exercise makes me happy. That's what the brain does. So the question is, is there any trigger between the brain and the actual the cells of the bone or cartilage to improve the regeneration? Do either of you have a better answer than that?
Yeah, that is a tough question. So, I mean, there would be two types of responses, right? So you could imagine systemic responses, and I think that's what you're trying to get at, is that somehow the brain, hormonal responses, there are things like parathyroid hormones, things that stimulate bone growth. But then there can also be the local effects, right? The fact that the cells, as Chelsea was talking about, the cells are sensing something about their environment, right? And that environment is telling them to do something. It's telling them to either grow new bone or resorb bone. And as Safa had mentioned as well, there's a pretty strong response there. And it doesn't need to be driven by the brain itself. It can be something that cells do autonomously by themselves because they have a program built into them that tells them what to do in response to mechanical stimuli. Thank you.
Oh, wait a minute. Actually, I have one more addition to that. So it also is I just wanted to highlight one of the studies we're doing right now at in the lab that may provide more insight into that. So one of the very interesting things is that if you hit your head in a traumatic brain injury and then you break your leg at the same time, which is often what happens in traumatic injuries, the bone, clinically, there's this phenomenon that your bone heals faster. So what we're trying to do is actually understand if there is maybe not a humoral response, but a direct neuronal response of the brain to the bone. And we have been investigating that, and one of the things we're seeing is that that we were wondering if, we still don't have the answer, but it is an area we're actively looking at, and we're trying to answer that question of, is there true biology behind this idea that if you damage your head and break your bone at the same time, that that your bone will heal faster? Because what we don't want you to do is go hit your head after you break your bone to make it heal faster.
That's not a good translational. And there's one other thing I had mentioned, another study that it's similar to what some other people have asked, and that they've done studies where they just look at exercise, say, for example, in sheep that are moving and just looking at bone growth. What they find is that bone mineral density will actually change in response throughout the whole body, not just in the locations they're being loaded. So there seems to be a mixture of both local and systemic responses, too.
Separate question, if I may. Have any of these techniques for building scaffolding and so on been patented?
Oh, yes. Yeah, lots of patents on that. And actually, it's a good and bad thing. But generally, the patents have, there's still a lot of space to patent these, and that's really one of the keys, is now the university has become active in trying to create patents, because what we need for these technologies to get out of the lab and into clinical care is for industry to take them the next step. Really, we can get them so far. We can do the animal studies, but universities, we're funded by the NIH. We're really into discovery, and so we can really discover the technology, and then we need industry to take them. And interestingly, without a patent, that really isn't valuable to industry. So it's a key to translating it.